Okay, so this is all about the lecture video on electroporation, a gene delivery method that we can use to transfer plant cell, animal cell and bacteria. So first we will learn about the instrumentation. So here in between a positive and negative pole there is a conductive suspended solution, and in that conductive suspended solution we have DNA along with our cell. Okay, that means let me explain the entire situation. The cell here is suspended in that conductive solution, and that conductive solution contains DNA. Okay, so in terms of gene therapy, the DNAs are our therapeutic genes. Okay, so now as initially entire cell is totally sealed, it is no near about impossible for the DNA to insert or to enter inside the cell. Okay, so now we will take the help of electrical impulse to create pores via which the DNA can insert inside the cell. Okay, so how those kind of pores can be generated? So let me told you that for that purpose we will use electrical impulse. Okay, a electrical impulse of optimum voltage for a very small amount of time, for let's say few microsecond to millisecond, we will apply through the solution. Okay, and as a result what will happen? So remember it very carefully that cell membrane has phospholipid bilayer, and as we have applied electrical impulse, so those electrical impulse will cause a disturbance inside in the phospholipid bilayer, and which facilitates the formation of the pore. Okay, so let's see this line, that an electrical charge at an optimized voltage and only a lasting a few microsecond to millisecond is discharged through the cell suspension. Okay, which will cause disturbance in the phospholipid bilayer of the membrane and result in the formation of temporary pores. Okay, so we will rise the amount of the electrical potential for a certain amount of time, so that the pore should be large enough to allow the insertion of the DNA. Okay, and after that, once the DNA is successfully entrapped, we will drop the voltage, which will causes the resealing of the pore. Okay, as you can see. Okay, first of all we will rise the electrical impulse or electrical potential, which will facilitate the formation of pore. Then we will drop the voltage, which will cause the resealing. Okay, as a result the DNA gets entrapped inside the cell. Right now, by appropriate machinery, if the DNA also contains vector, then inside the vector it should also have origin of replication stuff. So with the help of those kind of thing, it will also start its own replication, transcription and translation. Right, so that is the idea of electroporation. Means here we are using electrical impulse to create an electrical field. Right, now it is the question, okay, it is clear that the cell the electrical impulse creating a pore in the cell membrane, right? But how DNA get inserted inside the cell? Is DNA have some legs? No. So how? So let me told you, we are here using conductive solution. Okay, so for that reason, if you have basic knowledge about electrophoresis, then I think you can understand that as DNA is negatively charged due to the phosphodiester bond that it have, it will migrate towards the positive pole. Right, so as a result, the DNA molecules that are present in this side will try to migrate toward this side. Right, so they will get the chance to be inserted inside the cell. Okay, So that is the idea for the electroporation technique. So what are the advantage of the electroporation technique? So you can use our electroporation technique to transform any kind of cell types. Okay. And another important thing, that if you are willing to perform a transformation step in very small scale and you have a very small amount of sample, then electroporation method still can help you to get a transform cell. Okay. And another important thing, now let's discuss about application. DNA transfection and transformation, direct transfer of the plasmid in between cells, and gene transfer to a wide range of tissues. Okay. So now disadvantage, as it is very clear that we are using a electrical impulse, there is a chance that the cell membrane will be disrupted permanently. As a result, it will facilitate the cell damage. Okay. And it is possible mostly when we are using a irregular intensity pulse. Okay. Another important limitation, as here we are doing non-specific transport, right? So there can be a chance of ionic imbalance which will causes the death of the cell. Okay. So in electroporation, the application purpose is DNA transfection and transduction, along with we can transfer the direct causes, we can perform direct transfer of plasmid from one cell to another cell. Advantage are, so we can use the DNA electroporation or normal electroporation for transferring of any gene, and it will be transferred with high efficiency and sensitivity. Okay. But and also it will help you to perform your experiment with a small amount of sample only, but it has the disadvantage that it is non-specific. Okay. And that in turn causes ionic imbalance and in turn cell death. And if you are using irregular intensity pulse, then there is a chance of disruption of the cell or cell damage. Okay. Which can cause problem. Okay. So that was the concept of the our electroporation technique that is used for transfer of the cell in plant, animals and bacteria. Okay.